We've heard a lot about all of the really phenomenal team science that has gone on in some of the earliest AMP programs. And I wanted to take an adjacent space and really start to get some inputs and discussion around what the impacts of all of that great science and data and research has been to the communities that the AMP program is really designed to benefit: the patients, the researchers, the clinicians. And so I am very pleased to be joined for this final panel by the NIH Institute directors who have overseen the first 4 AMP programs, as well as 2 of our very many esteemed industry partners. So following the same order as our earlier scientific sessions, we'll start with Dr. Richard Hodis. Dr. Hodis has been the director of the National Institute on Aging since 1993. Which has led the AMP Alzheimer's disease projects, both the first iteration and second iteration, since the very first day of the AMP program. So Dr. Hodis, could you start by telling us, from your perspective, what has been the impacts of those 10 years of AMP-AD research?
Thank you, Courtney. Well, let me start with a little description of where we were in the beginning, what's happened since. So 2014, we all understood the severity of the challenge of Alzheimer's disease. We just had the beginnings of ways in which risk factors such as genetic ones could be applied. We had some of the early biomarkers. When we started in 2014, with the advantage of having worked with FNIH on ADNI, you've heard this referred to by David and Francis earlier, the Alzheimer's Disease Neuroimaging Initiative. I remember when this began, 2004, the question was, how could you have a program in which you're asking pharmaceutical companies to contribute funds when the results were going to be made available to everybody, and if they didn't contribute, they'd get the same outcomes. But lo and behold, it worked. So we've had the good fortune of experience with these partnerships, and certainly we've seen them perpetuated. So what's happened over that time? Well, first the partnerships went from co-funding, public-private, to true partnerships, scientific, intellectual, as well as financial. And this was reflected in some of the things you heard again mentioned this morning. So when we had early discovery of the pathways involved in Alzheimer's disease through AMP-AD, we had groups of public academics and private sector scientists collectively coming up with their consensus of what they thought were the best candidates and the most druggable candidates. And the impacts were real and immediate. So the drug companies reported back to us as outcomes that this led them to pursue interest in some, discontinue those, some of those they were active in, really then changing the trajectory of private sector. For us, it led to a point where now at NIA, we have some more than 60 ongoing clinical trials with a diversity of targets we couldn't have imagined before, not just amyloid and tau, which were common to us, but a whole new generation which are on the verge of making a real difference to the public, as we've discussed. I also would be remiss if I didn't mention the changes that have happened in the collaborations within academia. So we began in AMP-AD with a group of outstanding, brilliant investigators, some of them in the room, who were used to functioning actively, intensively, and effectively in relative isolation. You know, we would sometimes have meetings that brought all of our grantees together, but the notion that data would become available, transparent, open to the full research community was new at the time. Now, as we've heard, signs of success, including those, the fact that some of the greatest discoveries are coming from people not even funded through AMP, but as a result of these outcomes. So to the answer of where we are now, as you may know, there's been a lot of publicity about the first effective treatments of Alzheimer's disease. Effect was limited, but is the first breakthrough. 27% slowing has been quoted. But interesting, some of the recent data suggests there in terms of what we've learned from AMP-AD, if you break down further by biomarkers into the state of tau, for example, distinguishing the relatively early and late cases of Alzheimer's, for those with low tau translates into possibly early stage of disease, an impact of some 40% in slowing. So we have first breakthroughs, but I would say the real impact on the verge of coming to the public now is our ability to engage them in studies, in trials with biomarkers and a variety of targets that couldn't have been imagined through the great revolution in the spirit of open science and collaboration, public-private and public-public. And for this, we're very grateful. It couldn't have been done without FNIH, and the years have been truly remarkable for us.
Thank you for that. I'll turn next to Dr. Lindsay Criswell, who became the director of the National Institute on Arthritis and Musculoskeletal and Skin Diseases in 2021 after serving as the Vice Chancellor of Research at UCSF. So NIAMS has overseen the efforts of both the original AMP program in rheumatoid arthritis and lupus, as well as the expansion of the program to other autoimmune and immune-mediated diseases in 2021. So from your perspective, how has the work of both programs impacted the research and the patient communities?
So thank you, Courtney. And first of all, thanks to the FNIH and my colleagues, my fellow panelists, and the partners for organizing this symposium. It's quite thrilling to hear all of the discoveries and all of the progress that's been made over the past 10 years. And it's a privilege for me to be part of celebrating the successes. So, and speaking from the RA and lupus community, we were thrilled that RA Lupus was one of the initial programs, and absolutely, as you've been hearing today, this program has impacted how we do science and how we care for patients. And I can't possibly summarize all of the exciting results that you heard this morning, but I'm going to highlight just a few points in these areas, starting with some scientific accomplishments. The AMP RA Lupus program started in 2014, and to the credit of the teams involved, they pivoted relatively early to try to focus on novel technologies to dissect the molecular basis of these diseases, which continue to be mysterious and for which we had very limited understanding. And I know that was a very heavy lift to try to employ the single-cell technologies, but that has really turned out to be critically important in understanding disease mechanisms. You also heard this morning, and as recently reported in the journal Nature, that AMP investigators were able to study over 300,000 cells from synovial tissue, and they identified 6 major types of inflammation in rheumatoid arthritis, which they're calling cell type abundance phenotypes. And interestingly and exciting, these phenotypes appear to be predictive of treatment responses. So a really important development for rheumatoid arthritis. On the lupus side, the AMP team has defined the immune cell landscape in kidneys beyond what we, well beyond what we understood before. And they also found that gene expression of immune cells in urine and kidney was highly correlated. And this is a critically important development because it suggests that we may be able to use the urine as a surrogate for kidney biopsy. So that's a really important discovery that was made in the context of AMP. So what about ways in which we do science? I think beyond some of the technologies that have been employed in AMP, the AMP really deserves a lot of credit for some of the innovative processes for team science government and decision making. I think that's been critically important to the success. I've mentioned already the implementation of novel approaches for precision medicine and disease deconstruction. And along with those technical developments, the RA Lupus team also deserves a lot of credit for some of the computational strategies and the tools that they've developed to help analyze and interpret these massive, large-scale, single-cell data sets. So I think that's also, and of course, much of the progress in the AMP RA Lupus program we've been seeing across the AMP programs. They also were able to assemble a comprehensive data set using a wide range of molecular technologies applied to small tissue biopsies as well as urine and blood. This enables, of course, sharing of data, critically important to accelerate progress. And also, they developed, and this process enabled the development of standard protocols that has been shared across study sites and beyond the AMP, also critically important. We also heard this morning about the knowledge portal where data are being deposited for access not only by AMP investigators but by the broader research community, and this will also help to stimulate further research to leverage these important investments. I had the opportunity to participate in the AMP Lessons Learned working group, and one of the things that really struck us was the extent to which this massive, large, complex team science effort helped to launch careers of early career investigators, and that is something that wasn't necessarily anticipated and wasn't the sort of traditional advice you'd offer to an early career investigator. So what about some of the ways in which the program has improved patient care and treatment outcomes? Clearly, by understanding the disease better, we are getting a handle on new targets and potential approaches to treatments, and I've been really pleased to hear from some of the industry partners at this meeting that indeed, as a result of AMP, some of their efforts internally to prioritize or deprioritize targets has been accelerated. I mentioned also this pairing of blood, urine, and tissue samples. That will also pave the way for non-invasive diagnosis of disease, better monitoring of treatment response, and facilitate early clinical decisions. That's clearly something that's going to help patient care. The availability of synovial biopsies in the U.S. was very limited at the time this program was started. We heard how we were able to leverage expertise outside of the U.S. to bring synovial biopsies into the research realm in the U.S. That's changing the way we do science and facilitating our work. Also wanted to highlight that in addition to the identification of CD6 and IL-16 as potential targets and biomarkers for lupus nephritis, I think also important was the point that we heard from, I think, Dr. Bayan this morning, that we now understand that we should probably be biopsying kidneys in lupus patients at much lower thresholds than was traditionally done in the past. So looking forward, we were able to build upon the success of the RA Lupus program to expand to other autoimmune diseases, to leverage other emerging technologies such as spatial transcriptomics, to allow us to build from this kind of deconstructed status where we are understanding targets and molecular bases of disease to try to understand how cells and immune cells are interacting with cells in the tissues to cause inflammation and to further accelerate the identification of new targets. And lastly, I want to mention a couple of new efforts that have built upon the AMP RA Lupus program. I mentioned the benefit to early career investigators. We are so fortunate that thanks to the Office of Research in Women's Health, some support from NIAMS and the Office of Data Science Strategy, we now have a team science leadership scholar program that's been embedded within the AMP so that we can train additional people to learn how to be good team science leaders and mentors to expand this approach across our research moving forward. And lastly, as we'll hear more about tomorrow, I'm very excited about the AMP Systems Biology of Inflammation effort. This will build upon the observations that have been touched upon this morning about great similarities between seemingly very unrelated diseases. So that's a very exciting observation, and the systems biology of inflammation approach will allow us really focus on some of those unexpected discoveries in a much more powerful way and also support our data sharing efforts. So on that note, I'll pass it along to my next colleague here.
Thank you. And just to note, we have an incomplete data set, but at least 15% of the AMP-funded investigators are early career scientists. So certainly something that we could do better on, but are still very proud about that fact. So very similarly, the AMP Type 2 Diabetes program expanded in 2021 to a second project that was broadly focused on a number of different common metabolic diseases. And Dr. Griffin Rogers is the director of the National Institute of Diabetes and Digestive and Kidney Diseases, which has led both efforts. He's been in the highest levels of NIDDK leadership since 2001 and has served as director since 2007. So Dr. Rogers, how have the AMP efforts in your programs impacted the research and treatment of course, type 2 diabetes, but also looking forward into other diseases as well?
Absolutely. Well, thank you, Courtney. I want to start off by saying that I'm honored to have this opportunity to reflect back to a little bit about the history of the AMP projects and what we've achieved together. When AMP launched in 2014, the unique Type 2D partnership felt like a redefined high-risk, high-reward project. It was really unprecedented in its scale with diverse partners, each bringing their own audience, their processes, and their resources. And yet we welcome the chance to work in concert, sharing tools and trust as we departed so radically from business as usual. The excitement of the project was almost palpable. Of course, you heard from Dr. Collins that AMP was launched at a press conference at the National Press Club, and it was covered by international media. And this AMP T2D partnership involved 5 pharmaceutical companies, NIDDK, FNIH, and major patient organizations. and the academic industry collaboration we set out to create would collect and use the massive amounts of human genetic data generated by type 2 diabetes and leverage that information to prioritize targets for drug development and deepen our understanding of the causes of type 2 diabetes and its complications. At the time, this is circa 2014, there were around 400 million people worldwide who had diabetes, including 26 million living in the United States, and there was about 79 million Americans who were on the verge of type 2 diabetes, a condition of so-called prediabetes. Now, the diabetes cost at that time was estimated to be about $250 billion a year. Now, these numbers were, and unfortunately still are, stark, but they drove us toward a clear shared goal of making real and really fast progress towards the treatment of type 2 diabetes. And NIDDK and other funding agencies in the diabetes research community have long worked together understanding the genetic characteristics of individuals that could make them either more susceptible to the disease or protect them from it. And so type 2 diabetes genetic data became to accumulate more and more quickly during the first decade of this century. Now, in the early 2010, we saw an explosion of the discovery in the field, courtesy of the NIDDK's type 2 diabetes genetic exploration for next generation sequencing in multi-ethnic samples, or T2D genes, because of course we love acronyms. This project injected a vast quantity of genetic data with the potential to supercharge our understanding of T2D etiology, and yet realizing the potential would require widespread researchers' access not only to the T2D genes data, but also to those from other sources. And at the time, this effectively meant making requests and negotiating agreements with researchers responsible for each data set. And AMP circumvented this administrative burden. We created opportunities to jumpstart diabetes drug development by aggregating the data from those sources and providing powerful new tools to analyze them through the knowledge portal that you've heard about. And it was really through painstaking negotiations, AMP T2D brought existing data sets together. While ensuring patient privacy, this partnership provided the infrastructure and support to combine and harmonize more than 300 separate data sets from research studies around the world. And from the beginning, we worked to identify gaps in the existing data, and through continued research in partnerships and understudied communities, the project sought to plug those holes in the knowledge base. Now, a key hope of the knowledge portal was to drive innovation and speed the discovery process to identify and prioritize therapeutic targets. And gratefully, the pharmaceutical industry have embraced this resource, saving time and money spent in failed drug development. You heard a little bit about this from Dr. Otis. And they've invested some of that saving in helping make the knowledge portal still more valuable. And back when this began, I remember telling a reporter that we aimed to create a searchable library of at least 100,000 genetic samples. Now, 10 years later, that 100,000 seems rather humorous, understatement. By the time it expanded to include now, it's sort of evolved into AMP common metabolic diseases. The portal contains integrated, anonymized clinical and genomic data from more than 1.5 million people, along with ever-expanding suite of powerful analytic tools for researchers. And most importantly, the partnership continues investment means that the knowledge portal is continuing to evolve and continuing to catalyze research towards better treatment and outcomes of people with type 2 diabetes and for a host of other diseases and conditions as well. And what we began as a Type 2D knowledge portal was so successful in providing publicly accessible databases and analytic tools that its models was soon adapted to build knowledge portals for other complex diseases, many of which were integrated 3 years ago to create this AMP Common Metabolic Disease Portal. So thus, AMP established a new paradigm for scientific data sharing to allow researchers around the world to access a trove of data to test novel hypotheses and to generate new questions about how to prevent and treat complex diseases. And AMP CMD continues raising the bar for combining vast quantities of data, cutting-edge analytics, and making them free and open to the research community. And I would say importantly, AMP2D exceeded all the goals with respect to the identification, prioritization, deprioritization of targets in the pharmaceutical pipeline. And I believe this all goes to show that, when you get the right people in the room, what happens can truly be transformative. So at NIDDK, we feel fortunate to have had the opportunity to be involved in AMP since its inception. And I think I speak for all of my colleagues, and I say that we can hardly wait to see what happens next. Thanks, Courtney.
Thank you. And as we just heard in the last panel, the AMP Parkinson's disease effort is led by the National Institute of Neurological Disorders and Stroke under the leadership of Dr. Walter Koroshetz. Dr. Koroshetz, you became director in 2015, and the AMP PD program launched just 2 years later, so maybe that wasn't a coincidence. Even better, you're about to build on the success of that first program with the launch of an effort in Parkinson's disease and related disorders later this year. So can you tell us about the first program's impacts on the Parkinson's disease community?
Sure, Courtney. I'll make some of the same type of general comments that were previously made by the directors, maybe with a little bit of different flavor to them. I'd say, the first thing in my mind is that the AMP programs, and we have one in Parkinson's, we're trying to develop one now in amyotrophic lateral sclerosis. The real value is that as an NIH person, you're sitting down, you're trying to figure out how to get treatments for people with diseases, and you're talking to academics, you're talking to people with the disease, but the treatments have to come from the industry. And AMP is really a unique platform at NIH for actually to develop that community, that tripartite community, and that's what's been really valuable. I call it reality testing. If you have really great ideas and industry's not interested, there's something wrong with those great ideas, or you need a little more work. But I think that's the one thing I'd like to say, that the platform of AMP has been really valuable in the Parkinson's community, bringing together industry. FDA has also been very helpful there, and the Michael J. Fox Foundation and ASAP. And it's a democratic process. So I was in this room, how many years ago? I don't want to say. And we were planning the Parkinson's disease AMP program. We wrote everything down. I'm not sure we did what we said we were going to do, because it went in different directions. And it went in those directions because everybody has a vote. And you go along with what is the result of the vote. And that's just been a very revealing process. You really understand what is important to the people who are going to commercialize products to help our patients. That's the first thing. The second thing I think I wanted to state is that, I think everybody's mentioned this. We've seen this data that's just absolutely amazing from all these different AMP projects. But then I think about, when I saw patients, you're talking about tens of thousands of patients. I mean, it's enormous kind of effort that went on the part of the investigators, but also the subjects. And the subjects and the investigators did this because they wanted to advance the science. So the last thing that should happen in such an effort is for that data to be hidden in a file cabinet somewhere. So building one-stop shopping where people come into a portal like you've seen for these various AMPs and we have for AMP-PD is really, I think, just doing justice to the people who contributed their samples, their clinical data, and in our instance, it's their spinal fluid. which is really, really important, really hard to get, but you don't want, that's not the kind of thing you want to hide somewhere. You really want that to advance all of science, and that's what AMP PD has allowed us to do. Now, in our space, in Richard's space, you know, the brain is not accessible to biopsy. So we need something that we can track as the pathology starts and goes across, and we have a sense of that progression. Because with any treatment, that's what we call disease-modifying treatment, we're trying to stop that progression. But we can't do it by biopsying the brain. We need that liquid materials. And that's where I think the big emphasis has been in AMP. That's where the industry, once they see a path forward where these kind of biomarkers can inform whether their drug is hitting the target, that's going to really open the door to many, many different companies coming into the field. And so that's kind of been the goal for us at AMP, is to work with the industry to develop those kind of biomarkers that can attract them, that they can then go talk to their finance people and say, I think we can do this now because we have X, Y, or Z. And that's really the goal of our AMPs. We've gotten a lot from the genetics, certainly to open the door for these diseases in Parkinson's and ALS. But our big challenge is going to be branching the knowledge that we get from the genetic forms to what we call the sporadic forms. And there's been some progress there. Certainly, synuclein mutations were found in a family in which there was inherited Parkinson's, and now it is seen in 96% of people who have Parkinson's disease. There's now a method where in the spinal fluid with the seeding assay, you can make accurate diagnoses of a synucleinopathy inside the brain. And not only that, but those abnormalities become abnormal probably a decade before the motor signs of Parkinson's actually even appear. So we have this ability now with these biomarkers to actually diagnose people before they have serious disability, which is a great challenge because people are going to know that they're going to be getting this disease, and the pressure is going to be on us to bring new treatments. But we have things now that we can measure to see if our interventions are working. So I think it's really a bright future for these diseases where it was pretty dark in the past. And as you know, the unfair question that Deb Babcock got, it's true. We've made symptomatic improvements with our treatments, but we have not been able to modify the disease progression, and that's really the goal. And the last thing I'm going to just throw out is that looking to the future, there's another whole field that is emerging that I think is going to have a big impact, and that is the field that's been coming out of the Brain Initiative, which is focused on mapping, monitoring, and modulating brain circuits. And the reason why I think that's going to be important is because these liquid biopsies are important when they affect the function of brain circuits. So the patient doesn't care how much synuclein they have, they want to know whether they can turn and walk. And those are the circuits that are now going to be accessible to measurements. And also, the Brain Initiative is developing ways in which we can get genomic access to cell type specific, in a cell type specific manner. I think that's going to also open up a whole new area of therapeutics. So I think what we're doing here now on the molecular space is going to integrate with this circuit space in the near future, and then things are really going to explode. So a lot of optimism going forward, but this is all hard work, and one can't be one can't just dream. One really has to sit down with all those involved and really develop a path forward, and that's what the AMPs have allowed us to do. Thanks.
Thank you. So as we heard this morning and in some of the comments reflected just now, one of the core tenets of the AMP program is really the shared governance model and support from both the NIH and the private sector. I'm very pleased to have two of our private sector partners here to join the discussion today. So first, Dr. Melissa Thomas is a Vice President Medical for Diabetes, Obesity, and Complications Research at Eli Lilly and Company and served as the industry co-chair of the AMP Type 2 Diabetes program. Dr. Thomas, Eli Lilly has been involved with the AMP program since the launch in 2014, not just in the Type 2 Diabetes and its follow-on effort in common metabolic diseases, but also as a part of the first Alzheimer's disease program as well. Can you tell us from your perspective, what is the value of industry participation in the AMP program, and how have you seen the impacts of the research and the data?
Yes, well, thank you for the opportunity to be here today, and also what a pleasure to talk about AMP, particularly type 2 diabetes and common metabolic diseases with Dr. Rogers. And to reflect back on the beginning of AMP, as Francis Collins and David Woolley articulated so beautifully this morning, the beginning of the program, I was on the Target Validation Consortium as a representative from Lilly to be in the room with Boston Consulting Group and many other industry leaders, some of whom invested in AMP T2D and some of whom did not, but all who were in the conversations with some key academic investigators about where the gaps were and how we were going to build new drugs for type 2 diabetes. And we really did vet lots of different types of projects. It wasn't just genetics. We had several metrics we discussed. One was what was appropriate for pre-competitive space. Two, how hard it was to connect preclinical models to human disease, and how many failures there were in preclinical discovery and clinical investigation in trying to really transform care for people living with type 2 diabetes or complications. And so there was a sense of both optimism and magnitude of the challenge in designing and ranking the program that ultimately became AMP T2D. And from the industry side, you had to believe that we were going on an adventure in big team science that was going to raise the bar of disease understanding for all of us. And that was really important to us from the Lilly perspective, because we understood deconstructing or deconvoluting disease was a task too big for a single company or a single academic laboratory, but it was essential to connect what we could learn, particularly from human samples and people living with diseases, to how we prioritize drugs in industry. And so by nature, we were looking for bold projects that could be complementary across public and private sectors, and some level of optimism, a bit like Field of Dreams, that we would have a lot of building to do, but that at the end of the day, we would all understand more about human disease that would help us make better decisions about where to invest resources for what molecules to build. We had some of us, a few years of experience with an experiment in Europe that was the Innovative Medicines Initiative. So a few of us in the room from different companies had been in some big team science ventures that were still early, not delivering yet, but it gave us a reason to believe we could go there. And so at the end of the day, as we prioritized projects, there was a recognition that with all of the investments NIH had made and other sources around the world in generating more and more genetic data related to type 2 diabetes, there was still this big gap in understanding from those data what we should be prioritizing as targets. And the data were largely segregated, even though in individual communities and individual cohorts they had been highly collaborative. For a public investigator, a graduate student trying to decide what gene in a pathway should I make my knockout mouse for. The information there of what was connected to disease in humans versus how many papers they'd read, it was a gap. And so we intentionally, in designing AMP T2D, had several principles in mind. One, we wanted to make the data and the information and the understanding publicly available. That meant throughout public and private sectors. It meant sharing of data and information, not having a barrier where you'd have to pay for or have some special set of contracts to be able to ask questions of the data, but to democratize it so that all of that investment that the patients had made and that the funding institutions and governments had made to generate the information could move to understanding and then accessibility. It never would have happened without the amazing collaborative community in the type 2 diabetes genetics space. These are, and at the time were, amazing collaborative investigators. And it took a lot, as Dr. Rogers talked about, to connect the data sets and the information. But we also had to have early adopters. Not all investigators were comfortable sharing data. Not all were comfortable with the concept of finding insights and making them publicly available. But with early adopters and those who did believe, we were able to grow the resources. From the industry side, we could shape the program with our colleagues from NIH, with our colleagues in academia, and with tremendous support from the FNIH in those key crucial conversations about how to shape the research plan. We could, as Dr. Korshets mentioned, adapt as the science was adapting so that the methods and approaches we use, the resources changed as the program was growing. But I can remember about halfway through beginning to feel inflection points where we had been bringing data together, trying to get more and more data, more and more cohorts, and we had been building some analytic tools, sometimes when the data weren't really available to show what the tools could do. And we had an inflection point about halfway through when all of that building of analytic tools and data was coming together. And in one beautiful program meeting in conjunction with the American Diabetes Association, we had investigators who had brought their data sets into the portal, and they knew their data inside and out, and they were world experts, and they sat with our tools and looked up and said, I've never seen this before. I can see things in my data set that I would never have looked at when we had the more traditional siloed approach of a team with the data. It is the power of big data that individual investigators, whether they're in public or private sector, are going to view it differently, ask questions differently. And by building this resource, we were able to provide so much more value than we ever could in individual projects alone. And so it's been a tremendous experience, the collegiality across different companies, across FNIH, the investigators. And really, as the science and the understanding of where we needed to go in building new medicines for people living with common metabolic diseases was telling us that for the next generation project, it would need to go beyond type 2 diabetes. It was the strength and the innovation of the knowledge portal and what we had built in AMP T2D that made us believe that we could then cross interdisciplinary boundaries of where typical investigation areas sit and build the project for AMP common metabolic diseases where we can ask questions across cardiovascular disease and diabetes, across obesity and kidney disease. There are ways in which what we've been building allows us to ask questions relatively rapidly, at least for hypothesis generation, at least for the first step, that helps us prioritize how to spend our time, our resources, and we hope how we get to transformative new medicines for people that really intercept these diseases much earlier and give them healthier, longer lives. So what a pleasure to work in this endeavor.
Thank you. And if we use Field of Dreams as the metric, I think the program has been a wild success because we built it and you all did come. So last but certainly not least, Dr. Maria Quinton is currently the Scientific Director of Precision Medicine Neuroscience at AbbVie and was previously a Scientific Director with Takeda. We've been very pleased to have the support of both AbbVie and Takeda throughout the life of the AMP program. And you were also a very early supporter of developing the concept for the Systems Biology of Inflammation initiative. So could you tell us a little bit about your perspectives on how the AMP program has had impacts, and maybe a little bit of what you saw the need for bringing together the data sets in AMP and how that could increase the impact?
Thank you, Courtney. And first of all, I want to thank you for inviting me to this panel. I'm very honored to be part of this with all these established scientists. And also, I love seeing all the AMPs together. So I've always been focused on neurodegeneration, so I've seen AMP-AD, I've seen AMP-PD, but seeing all the data that have been generated across all these therapeutic areas is absolutely amazing. So one of the major challenges in drug development, if not the challenge, is patient heterogeneity. And I think we all know now, especially from today, it's not just in neurodegeneration, it's in every disease. And we're finally moving from having diagnoses based on clinical presentation to diagnoses based on biomarkers. But we need more. We need to really start going toward more precision medicine in order to address this heterogeneity. And we cannot do that without all the data that are being gathered through all these AMP programs. So in order to address this, we have to generate multi-omics data. We have to look at multiple diseases. And seeing what has been done to date is absolutely amazing. We have data sets from multimodal types of data. We have genomics, epigenomics, transcriptomics, proteomics, metabolomics, all of that across multiple matrices, and that's really important, especially in neurodegeneration where we can't do brain biopsies. Even CSF samples are not always easy to get, especially if we want to move to earlier diagnosis where we're not going to see people that aren't even patients yet and say, hey, give us a CSF sample so we can tell you what you're going to develop in the future. So having these data sets developed across matrices, we have plasma, we have CSF, we have brain data, synovial fluid, the fatty acid, we have so many different matrices available to really put all these data together. We have deeply phenotyped individuals with clinical data across platforms. So you saw earlier today, even comparison between targeted proteomics, Olink panels, SomaLogic. All these data help people make decisions and help really understand what's a real finding and what maybe not so real, and across cohorts. And I think that's also something really important, because something we can do on our own is get some samples from a specific cohort, we get data, but we don't know if it's a real finding until it's been done multiple times across multiple cohorts. And finally, something that hasn't really been mentioned is adding multiple ethnicities as well. And we saw that with AMP-PD and AMP-AD. There are major differences between different ethnicities, and so far, a lot of medicines are being developed for Caucasians, right? So all these data sets are now coming together, and it's really allowing us to make a lot more informed decisions on what to target, what biomarkers can help us understand how to select our patients properly, understand how they're progressing in the disease, and we basically now have this access to all this amazing curated high quality data that we can use to make decisions. And something I've noticed is when I started on the AMPs, I think maybe it was 5-6 years ago, people were talking about the AMPs. Have you heard about this? They're generating data. And now it's to the point where when we have a question, whether it's a new target that gets published or a new potential biomarker, typically the first thing we do is, oh, let's go check the AMP-AD database or the AMP-PD database. So it's almost become like a reference book of all the data out there. And this would not have been possible without all of these initiatives. And I think it's easy to forget that these data didn't just appear. This is from all the work, from all these initiatives that resulted in these data sets. So to me, what I've seen is now we have this available. And maybe we're not there yet. We don't have the biomarker panel that we need to identify PD patients that are about to progress. But we're starting to see things progress. With Alzheimer's, you saw it earlier today, we're seeing different classes of patients that maybe don't present the same. Some of them have metabolomics differences, some of them have differences in their epigenetics. Surprisingly, proteins didn't seem like the thing that differentiates these different cohorts of individuals. But so we're starting to see patterns. With Parkinson's disease, we're starting to see novel biomarkers that we didn't really even think of, like dopamine decarboxylase. That came up. The first thought was, oh, it's drug-related. And it appears that maybe it's not. So we're really seeing all these new data now that we can probably take to the clinic now and really use to understand why certain patients respond, why certain patients progress. So I'm just in awe by all the data that have been generated. And now we're also talking about doing all of this across diseases. And that's the AMP-SBI, I think, is something that will really generate a lot of interesting hypotheses, hopefully that can be tested. But nobody just has one disease, right? So having the data across all of these is going to be extremely valuable. And perhaps this can enable basket trials in the future. So I'm just in awe by everything that's been generated and really, really happy to see all the progress that's been made over the last 10 years.
Well, thank you so much, Melissa and Maria, for those perspectives. So I want to circle back to my NIH colleagues on two things that came up in Melissa and Maria's comments. The first, Melissa, I love the comment of crossing interdisciplinary boundaries. And certainly AMP-SBI will hopefully lay the groundwork for that. But just even within the AMP Executive Committee, which you all participate in, can you talk briefly about what that participation has meant to you and how the integration of the data might help you broaden those opportunities?
I presume we'd all be happy to respond there. So just to point out the progression we've been talking about in terms of data and understanding of underlying biology, moving from the simplistic one disease at a time to the notion of complex processes, which will vary among individuals with a given diagnosis and maybe in common across individuals with different diagnoses. We've moved to the point where, in the area of Alzheimer's disease, but I'm sure in the other disciplines as well, we were aspiring to have a database in a single laboratory that if we were lucky, they were willing to share. And that was clearly just not going to do the job, so we moved towards having common databases in a portal that could access many. And when we did that for AMP, this was an important step. I think most of us then broadened it to all of the data that we have in a given disease area. And now the critical insight, which you've heard over and over again, is that we've got a lot to learn across diseases and these entities. So the notion that a single portal for Alzheimer's, a single portal for autoimmune diseases nominally is sufficient is now seems in retrospect naive. So we're going to be looking, I think, with a great excitement to the time when we can analyze individuals with a given clinical diagnosis for biomarkers, for multi-omic profiling, and find mechanisms that undoubtedly are going to be in common across diseases in ways we hadn't anticipated. Part of AMP-AD 2.0, in fact, we haven't talked as much about heterogeneity of populations, not all of us at least, including myself. If there's an area of failing, it's having failed to acquire data to date in a satisfactory way for the diverse populations. And this is going to be yet another dimension of expansion. And I think we're all on a common page here and probably going to be doing this across diseases. So yes, share the excitement. A lot of these ideas for analysis across AMPs did occur at the executive and extended executive committee meetings. They were real and substantial, and those insights from leadership there have certainly affected me, and I suspect my colleagues as well.
Yeah, a great topic, and I guess I'll share back to this audience a phrase that we've talked about a lot, and that is the notion that we're moving towards this elusive molecular taxonomy of disease. So a concept that was developed quite a number of years ago, and we now seem to be just on the brink of really being able to get to that level of understanding. And many of us in the sessions earlier today really focused on the promise that that is going to provide and the kind of revolutionary impact that's going to have. I guess I'll also just add that my conversations and the discussions that I've been privileged to be part of also speaks to this notion of a research ecosystem for chronic disease, where you're not simply talking about different diseases that may be common or rare. You're talking about different types of multi-omics data, but you're also talking about the environment. You're talking about social determinants of health. You're talking about psychosocial factors. So I hope that we can also, building upon the AMP data and what we're learning, think even more expansively about some of the other factors that interact with the genome and that influence health, and be thinking about chronic disease, rare and common, and towards this elusive, deep understanding of what causes disease in humans.
Thanks. Let me sort of just riff off of something that Dr. Thomas just mentioned, and that is this notion of heterogeneity of diseases. And I think my colleagues have expressed the same idea when we went into this project related to AMP to sort of better clarify molecular, cellular, and omic data on individuals that had common diseases. It became clear that, while the knowledge portal clearly allowed us to aggregate a lot of data sets and to make reasonable hypotheses based upon, what we were learning, it also brought, into clear and sharp focus the notion that disease heterogeneity even exists among various groups. And so, what I would say is that, in developing this initiative, our institute is sort of, really making a big push now to develop a better understanding related to disease heterogeneity, including, not only, genomic, and transcriptomic, and proteomic, perhaps all omics just limited by economics, but, understanding, I'm here all week, understanding better the social determinants of health, as Dr. Criswell, and how that really impacts the disease progression, for example. Just getting back, though, to the knowledge portal, and again, this is something that Dr. Thomas mentioned, we were able to jumpstart this project by having studied for a number of years the genetics, particularly in U.S. population, but it really was putting this together and speaking with colleagues, in Europe and other places that allowed us to really make big breakthroughs, because we had to obviously abide by their principles, and developing a federated data system that we then had access to query those ideas and the hypotheses that came out really was really a big step in a direction. I know Melissa was there at those meetings, and it wasn't something that occurred overnight. It took, a lot of effort to sort of get people to sort of come in line to understand that the good of the many outweighed the good of the few. With respect to that portal, though, I would, just want to point out a fact that in the 6.5 years of this type 2 diabetes knowledge portal, allowed scientists to sort of analyze, and in that period of time, they were able to identify 6 new potentially druggable targets, to prioritize 18 targets, and perhaps most importantly, to deprioritize 30. And I think this is really the reason that industry was very much, very interested in it, because, you want to fail early. You want to not put additional funds into something that's not likely to be successful. Thanks.
Well, I'm going to say that you mentioned the word multidisciplinary, and I think AMP projects bring into focus the need to bring in computational scientists like we've never seen before. So, and I think hopefully the richness of the data will attract them, but I think that's right now our Achilles heel, that we we have these great data sets, they're very complex, and you can't look at them and something's going to jump out at you. You need really sophisticated tools that then can be primed by, as Griff said, association with things that don't work, things that do work. And so I think that's an evolving science that I think AMP can do like no other aspect at NIH.
Thank you. Well, we have just a few minutes left together, but I didn't want to wrap up without touching on one more thing that I think is critically important. And Maria, you alluded to this idea of expanding patient diversity within the clinical trial cohorts, within the data. And I know that the various AMPs have made some really great strides in that space. So briefly, if anyone would like to comment on how that has provided greater impacts to the patient communities.
Well, I can just share that lupus is a fantastic example where that's critically important because the disease is more common, it's more severe, poor outcomes in individuals from non-European ancestry backgrounds. And unfortunately, the early genetic studies and many clinical trials, et cetera, have not necessarily been representative. So I was really pleased to see the slides earlier today that showed the diversity of some of the cohorts for lupus, and I hope that we will be seeing that, as we also saw this morning, increasingly across all of these studies, because until we understand what's happening in the populations around the world, we won't have a full handle on disease.
Yeah, I was going to say that the talk that we heard about the studies in Nigeria for Parkinson's. So, is Mary still here? Okay. Oh, there she is. So my understanding is that this was a training exercise, that no one expected that they were going to find something. But the fact that they got such a big effect size, so we're all really happy about it, but it also brings home the point, like, what have we been doing for the last 20 years? We missed this like gigantic signal because we didn't look. I mean, I just wonder, where are these other signals out there that, they're just sitting there and we're just neglecting them? But is that correct about the training?
No, that's true. Okay. It is true. So we were getting people to get on the Terra platform. Like we said, everything was under the same platform. And this was really important because obviously we're really fortunate here and we're resourced appropriately, but obviously that's not the case in the rest of the world. And so when we started with this process, we were working ground up. These individuals, they had significant physician training, but that's about it. No genetics, no coding, no nothing. Cloud infrastructure, even if you are familiar with coding, it's still a little bit of an adjustment and it's a threshold to meet. So we started it as a training exercise, found something. We're convinced we were wrong, because like anyone can tell you here, you see something, it's almost too good to be true, you're skeptical. So it did initially start that way, but I'm glad we persevered. And I'm glad that we really wanted to put this in the hands of the people who had contributed the data, and they were the ones out on the field and on the ground. And I'm glad that they couldn't present it here today, so I'm presenting on their behalf, but that's how it started, yeah.
All right. Well, this has been absolutely wonderful to have such an immense amount of leadership and brain trust and initiative sharing the stage with me today. So thank you for all of your remarks and comments. Please join me in thanking our panelists today. And we just really appreciate the perspectives that you all have provided on how the scientific work of the AMP projects has led to and will continue to lead to really important benefits for researchers, for patients, and we're really excited to see how those impacts continue to grow over the future.